Hello Internet, our topic today is on understanding cell division regulation. In this video, we will explain the implications of cancer. If you are new here, welcome. We are a group of retired professors sharing bite-sized videos in life sciences. Let's dive into the topic for today. What is cell division? Cell division is a fundamental process in the life of organisms, as it allows for growth, repair, and reproduction. The two main types of cell division are mitosis and meiosis. Mitosis is the process by which a single cell divides into two identical daughter cells, while meiosis is the process by which a single cell divides into four genetically distinct daughter cells. Importance of cell division. The regulation of cell division is crucial for the proper functioning of living organisms, as it ensures that cells are produced at the right time, in the right place, and in the right numbers. This process is essential for the growth and repair of multicellular organisms and is tightly regulated to ensure proper cell function and maintenance of genetic integrity. The regulation of cell division involves a complex interplay between various molecular and cellular mechanisms that ensure the proper coordination of cell growth and division. Introducing mitosis. Mitosis is a complex process that is divided into several stages. The first stage is prophase, during which the chromatin condenses into visible chromosomes. Each chromosome consists of two identical sister chromatids that are held together by a centromere. The next stage is prometaphase, during which the nuclear envelope breaks down and the chromosomes attach to the spindle fibers that form the mitotic spindle. This is followed by metaphase, during which the chromosomes align at the center of the cell, or the metaphase plate. Anaphase is the next stage, during which the sister chromatids separate and are pulled towards opposite poles of the cell by the spindle fibers. Finally, telophase and cytokinesis mark the completion of mitosis, during which the chromosomes decondense and the cell divides into two identical daughter cells. Introducing meiosis. Meiosis is a more complex process than mitosis, as it involves two rounds of cell division. The first round, meiosis 1, is similar to mitosis in many ways, but it also involves a process called crossing over, during which genetic material is exchanged between homologous chromosomes. This results in genetic diversity among the daughter cells. The second round, meiosis 2, is similar to mitosis in that it results in the formation of four genetically distinct daughter cells. Cell cycle. Cell division is a tightly regulated process that is controlled by a variety of proteins and signaling pathways. One important protein that plays a key role in mitosis is cyclin-dependent kinase, CDK, which is a protein kinase that controls the progression of the cell cycle. Phases of cell cycle. The cell cycle, the series of events that occur in a cell leading to its division and duplication, replication, can be divided into two main stages: interphase, G1, G2, and the mitotic phase, EM. Interphase. During interphase, the cell grows and replicates its DNA in preparation for cell division. The G1 phase is the first stage of interphase, during which the cell carries out its normal functions. The S phase is the stage in which DNA replication occurs. Finally, the G2 phase is a short period of growth and cell checkpoint before the cell enters the mitotic phase. During the mitotic phase, the cell physically divides into two daughter cells. This process is called mitosis and includes stages such as prophase, prometaphase, metaphase, anaphase, and telophase. G1 phase. The G1 phase is the first stage of interphase, in which the cell grows and carries out normal functions. The S phase is the stage in which DNA replication occurs. The G2 phase is the final stage of interphase, in which the cell undergoes final growth and checks for any DNA damage before initiating cell division. G1 to S phase. The transition from G1 to S phase is regulated by the activity of cyclin-dependent kinases, CDKs, which are enzymes that control the progression of the cell cycle by phosphorylating specific target proteins. CDKs are activated by binding to cyclins, regulatory proteins that are synthesized and degraded in a cyclic manner. The cyclin CDK complex phosphorylates and inactivates the retinoblastoma protein, RB, which acts as a brake on the cell cycle by inhibiting the activity of E2F, a transcription factor that promotes the expression of genes necessary for DNA synthesis. G2 checkpoint. The G2 checkpoint, also known as the second growth checkpoint, is a critical stage in the cell cycle that occurs during interphase. It is the final checkpoint before the cell enters into the mitotic phase, where the cell physically divides into two daughter cells. What happens during G2 checkpoint? During the G2 checkpoint, the cell ensures that DNA replication is complete and that no errors have occurred. The cell also monitors the overall health and integrity of the cell, making sure that it is ready for mitosis. If any issues are detected, the cell may halt the cell cycle and repair the damage before continuing. The G2 checkpoint is controlled by a variety of proteins and signaling pathways, including cyclin-dependent kinases and checkpoint kinases. These proteins work together to ensure that the cell is prepared for mitosis and that the cell's DNA is intact before replication. Disruptions to the G2 checkpoint. Disruptions to the G2 checkpoint can lead to errors in cell division, which can result in chromosomal abnormalities and potentially cancerous growths. In addition, mutations in the genes involved in the G2 checkpoint can also lead to cancer. Therefore, the G2 checkpoint is a very important stage in the cell cycle, as it helps to ensure the proper replication and division of cells. Signaling integration. The cell cycle is tightly regulated and controlled by a variety of proteins and signaling pathways to ensure that proper replication and division occur. Any disruptions to these processes can lead to errors in cell division and potentially cancerous growths. Regulation of cell division. The regulation of cell division is a complex process that involves various signaling pathways, proteins, and enzymes. The main signaling pathways that regulate cell division are the mitogen-activated protein kinase, MEIPK, pathway, the phosphatidylinositol 3 kinase, PI3K, pathway, and the RAS/RAF pathway. These pathways are activated by growth factors, such as epidermal growth factor, EGF, and insulin, and are responsible for initiating cell division. MEIPK pathway. The MEIPK pathway is activated when growth factors bind to their receptors on the cell surface. This triggers a cascade of phosphorylation events that lead to the activation of the mitogen-activated protein kinase, MEIPK, family of enzymes. These enzymes then phosphorylate downstream targets, such as transcription factors, which then enter the nucleus and activate genes that promote cell division. PI3K pathway. The PI3K pathway is activated by growth factors that bind to their receptors on the cell surface. This triggers the activation of PI3K, which then phosphorylates phosphatidylinositol for 5-bisphosphate, PIP2, to form phosphatidylinositol 3,4,5-triphosphate, PIP3. PIP3 then binds to the protein kinase B, ACT, and the target of rapamycin, TR, proteins, which enter the nucleus and activate genes that promote cell division. The RAS/RAF pathway. The RAS/RAF pathway is a critical signaling pathway that regulates cell division and proliferation. It is activated by growth factors, such as epidermal growth factor, EGF, and fibroblast growth factor, FGF, and plays a key role in the regulation of cell proliferation and differentiation. The RAS/RAF pathway is also closely linked to the regulation of the cell cycle. The MEK/ERK pathway activates the transcription factor E2F, which promotes the transcription of genes required for DNA replication and cell division. Additionally, the RAS/RAF pathway can also regulate the activity of the cyclin-dependent kinase, CDK, family of proteins, which are key regulators of the cell cycle. RAS proteins. The RAS family of proteins, including KRAS, NRAS, and HRAS, act as molecular switches that transmit signals from the cell surface to the cell interior. They are activated by growth factor receptors, such as EGFR and FGFR, which bind to the extracellular domain of the receptor and activate the intracellular RAS protein. Once activated, RAS proteins bind to and activate RAF kinases, including ARAF, BRAF, and CRAF. RAF kinases then activate the downstream MEK/ERK pathway, which leads to the phosphorylation and activation of transcription factors, such as ELK1 and CFOS. These transcription factors then activate genes that promote cell proliferation and survival. Tight regulation is critical. The RAS/RAF pathway is tightly regulated to ensure proper cell division. Several negative feedback mechanisms, including the phosphatase PTN and the protein SPRY2, act to inhibit the activity of RAS and RAF proteins. Additionally, the RAS/RAF pathway is regulated by the tumor suppressor protein p53, which can inhibit cell proliferation and promote cell death in response to DNA damage. What happens when cell division goes wrong? Take a moment to think. Yes, you are right. Cancer is a disease characterized by the uncontrolled growth and spread of abnormal cells in the body. The hallmarks of cancer, a concept proposed by Douglas Hanahan and Robert Weinberg in 2000, describes the 6 key characteristics that distinguish cancer cells from normal cells. These hallmarks include the following, which will be described shortly. Self-sufficiency in growth signals. Cancer cells have the ability to grow and divide independently of normal growth-regulating signals. This allows them to evade the normal mechanisms that control cell division and growth. Insensitivity to growth inhibitory signals. Cancer cells also have the ability to ignore signals that would normally stop cell growth and division. This allows them to continue growing and dividing uncontrollably. Evasion of programmed cell death. Cancer cells can evade the normal process of programmed cell death, also known as apoptosis, which allows them to survive and accumulate in the body. Limitless replicative potential. Cancer cells have the ability to divide and replicate indefinitely, which leads to the formation of tumors and the spread of cancer throughout the body. Tissue invasion and metastasis. Cancer cells have the ability to invade and migrate to other tissues and organs, leading to the spread of cancer to distant parts of the body. Sustained angiogenesis. Cancer cells can create new blood vessels to support their growth and proliferation, which is known as angiogenesis. Other hallmarks. These hallmarks of cancer allow cancer cells to evade the body's normal mechanisms for controlling cell growth and division, leading to the formation of tumors and the spread of cancer throughout the body. In addition to these 6 hallmarks, researchers have also identified additional hallmarks of cancer, such as genetic instability and immune evasion. Genetic instability refers to the accumulation of mutations in cancer cells, which can lead to the development of new characteristics and abilities in cancer cells. Immune evasion refers to the ability of cancer cells to evade detection and destruction by the immune system. Closing perspectives. In summary, the hallmarks of cancer describe the key characteristics that distinguish cancer cells from normal cells. These hallmarks include self-sufficiency in growth signals, insensitivity to growth inhibitory signals, evasion of programmed cell death, limitless replicative potential, tissue invasion and metastasis, and sustained angiogenesis. Understanding these hallmarks is crucial for developing new treatments and therapies for cancer.